to talk about rapamycin, and I think it and its cognate rapalogs and TOR kinase inhibitors are one of the groups of chemicals that are furthest along the route to genuine translation into geroprotection. So rapamycin itself can extend lifespan in all the main model organisms, including mice. It can protect against various of the ills of old age in mice, for instance, cognitive decline, immune dysfunction, metabolic problems. And as Joan Manick has told us so elegantly, also it's showing great promise as a suppressor of immunosenescence in humans. So it really is an important and interesting drug. But with chronic administration, even at the low doses that are used for geroprotection, it can have side effects. So for instance, in mice, long-term use of the drug can induce insulin resistance. So we really need to understand better what are the optimal dosing regimes for the drug that minimize the dose that we have to use, and also what are the molecular mechanisms by which it's inducing geroprotection, because if we could perhaps target those more directly, we might be able to get the benefits without the costs. So we've been doing quite a bit of work on rapamycin in both Drosophila and in mice, and it's that I would like to talk about. So the drug does extend lifespan in Drosophila. This is work that was done some time ago by Ivana Bedov when she was in the lab in London. She has her own lab now. And you can see here the dose-dependent increase. It's not huge in Drosophila. The maximum lifespan extension is not enormous, but it's very highly consistent and reproducible. And because it was Drosophila, she was able to rapidly do quite a bit of specification of the nature of this lifespan extension, and specifically to do interaction tests with mutants to figure out which of the very many outputs of TOR signaling were involved in the lifespan extension. And what she found was that it was entirely TORC1 complex that was important. TORC2 played no role in the lifespan extension. The increase in autophagy and suppression of S6 kinase activity that happen with inhibition of TORC1 were both required for the lifespan extension, so if she blocked them genetically, she no longer saw it. And there was also a big sex difference. The lifespan extension was confined largely to females. And this involvement of the S6 kinase has been a long-standing mystery. It seems to be one that's conserved in mice. If you knock out S6 kinase 1 in mice, you get long-lived females. But the mechanisms are really unclear. And tomorrow there'll be a talk from Pingxi Yang from the Cologne Institute about the role of the S6 kinase. I think he's made great progress with understanding why its suppression is important. And there are also two posters from Yuxuan Lu, who's also here. The first one looking at the sex differences that I've mentioned, why the effect is confined to females, and also more on the memory effect that I'm about to talk about. So what I want to focus on is this, which is that there's quite a remarkable effect of really short-term treatment with rapamycin early in adulthood on what happens subsequently. So that was discovered by Paula Danzig, who's shown here when she was a graduate student, actually. It's been a long job, this one. There's a great deal of work by all four of these as postdocs to actually work out what's going on here. But the phenomenon is that if you feed the flies rapamycin for the first couple of weeks after they come out as adults, let them mature, and then you administer the drug, and then you withdraw the drug and wait and allow the flies to die, you see just as big a lifespan extension as you do with chronic treatment throughout adulthood. So that memory of the earlier treatment is carried right through to the time when death occurs. You get full protection. And this doesn't seem to be anything trivial, like them not clearing the drug, the signaling status not reversing, or some involvement of the microbiome. We've been able to eliminate all of those as possible explanations of what's going on here. And what we did was to exploit the sex difference, the fact that the drug only works in females, because that's associated with a very big difference in the way that the intestine ages in the two sexes. So you've got that here. I can't point to both sides at once, but the males at the bottom here getting older, going to the right, and you can see that that beautiful ordered columnar epithelium that the gut consists of in Drosophila is basically unchanged as they go through adulthood. It keeps its order and it doesn't become leaky. Whereas in the females, who you can see at the top, things are very different. Even in quite young females, you start to see proliferation of stem cells. Some of them misdifferentiate. You get dysplasia, disordered arrangement of the nuclei. The whole thing pulls away from the basement membrane. You start to see wound healing rosettes. And eventually the gut becomes really leaky. So there's great gut pathology in females as they age. And that pathology, all aspects of it, are completely rescued by rapamycin treatment. But interestingly, they're equally rescued just by this very early treatment. So I've shown two of them here. We've got dysplasia at the top on the left. You can see the control on the left there with the very disordered arrangement of the nuclei, which you don't see with chronic rapamycin treatment, or in flies that were treated for 2 weeks, looking very much later at day 60. So this is a really long memory effect. You can see the gut leakage at the bottom. We measured that with a blue dye that goes into the fly if the gut is leaking. And you can see the quantification on the right. Basically, both chronic and short-term produce an equal rescue of this gut pathology. Now, I mentioned that the increase in autophagy, macroautophagy, that happens as a result of TORC1 inhibition is required for the lifespan extension. And it also shows an associated effect of this early rapamycin treatment. So this is shown here measuring autophagy with LYSA tracker, simply looking for acidic vesicles. We can measure autophagy in many different ways, and they all tell the same story. So what you can see is the increase, so this is in the gut. With chronic rapamycin treatment, you get a big increase in acidic staining vesicles. But the same is true of the early treatment. If we look 10 days after the drug's withdrawn, all the signaling's gone back to normal, we still see this increased evidence of the vesicles, and it's shown at the bottom on the left. Now, that could mean that flux through the autophagy pathway has actually been blocked, or it could mean that it's been increased, depending exactly what's going on. And you can sort that out with the data on the bottom at the right, which is cyto-ID, which stains autophagosomes. And if the flux through the pathway is unaltered, the expression of that should be unaltered, and it is. So it looks as though there's increased flux through the autophagy pathway, and that it carries a memory of this earlier rapamycin treatment. And it turns out that that increase is causal in this memory effect. So we can block the increase in autophagy by doing RNA interference against one of its key components, ATG5. ATG5 is a very complicated molecule. It's involved in different stages of autophagy and different types of autophagy. But if we inhibit it, what we do is to completely block the beneficial effect of rapamycin on gut pathology. I'm not going to drown you in data, I won't show that, but this is the effect on lifespan. So if we look at chronic treatment on the left, what we're doing here is doing the RNA interference against ATG5 just in the enterocytes of the gut. So these are the big absorptive cells which make up most of the gut. And the interference itself doesn't do anything to lifespan. But what it does is to block the increase from the rapamycin treatment. And on the right, we've got the equivalent, but now for the short-term treatment. So both the RNAi and the drug are administered only for the first 2 weeks of adulthood, and you get exactly the same blocking effect. So if we stop that increase in autophagy that happens during the period that rapamycin is being administered, we no longer see the increase in lifespan. And we can do the experiment, or ask the question the other way around. If we increase macroautophagy, is that sufficient to recapitulate the effects of rapamycin? Again, I won't show you the results for gut pathology, but we get a complete rescue, both with chronic and brief induction of ATG1, which is what kicks off the classic macroautophagy pathway. And we get the same for lifespan. So just like the things I just showed you, we've got the controls. If we overexpress ATG1 chronically in the enterocytes, in the gut, then we see an increase in lifespan. And it's about the same as the increase that we get with rapamycin. And if we put them together, we don't see any further increase. And on the right, we have the same result for the brief treatment, exactly the same effect on lifespan. So we need this brief increase in autophagy in the first 2 weeks of adulthood to see the subsequent effects on the gut pathology and lifespan. And interestingly, what autophagy seems to do once it's been induced is to keep some form, at least, of autophagy running. Again, we can measure this in various ways. I'll show you the result for the fly equivalent of mammalian P62. So if you induce chronic autophagy in the mammalian kidney, for instance, then what you see long-term is a decline in P62. There's an initial increase, but with chronic treatment it goes down. And what you can see on the right is the quantification of the fly equivalent, which is called REF2P. And what you see is that with chronic rapamycin treatment, with brief rapamycin treatment, it's decreased, also with overexpression of HG1, and if you put them together, there's no further decrease. So something about autophagy is remembering this early autophagy in the first 2 weeks of adulthood. We've spent a great deal of time trying to understand what this autophagy memory is and how it's being induced. And we initially tackled it by simply doing a proteomic analysis on the intestine. So what we asked was, are there proteins that show persistent changes in expression that correspond to ones that we see with chronic rapamycin treatment, but that persist after the drug is withdrawn? And there weren't that many, actually, but for the ones that we did find, there was a very clear signal of two things. One was increased expression of proteins involved in branched-chain amino acid metabolism. I'm not going to have time to talk more about that, but there is a small story there. But also carbohydrate metabolism. And particularly what we saw was an increase in expression of lysosomal monosidases. And that really struck us because we've seen it before in the gut of long-lived insulin mutant flies. These monosidases come up. What they do is to remove mannose from carbohydrate moiety on glycosylated proteins, or glycoproteins rather. And it's a very important regulatory process in their catabolism. And actually quite a number of human genetic disorders are associated with lesions in the genes involved in that pathway. So it's one that's really capable of inducing cellular stress. And at least one of these lysosomal monosidases was clearly regulated at the RNA level. So we were able to manipulate it experimentally with RNA interference. This was LMAN5, and what we found was that if we did interference against it, again in the enterocytes, this blocks the long-term increase in lysotracker staining from a brief rapamycin treatment. So the pictures are shown on the left, the quantification. On the right, basically on its own, again, it doesn't do anything, but what it does is to block the increase with the 2-week treatment of rapamycin. So it's clearly a player in this whole causal pathway. And by the same token, if we briefly induce expression of it in enterocytes, it increases lysotracker puncti. So again, at this point, we still haven't nailed it. Clearly, this is a player, but what is it doing? And we were very struck by this lysosomal puncti, because at about the time that we were doing this, a paper came out about the gut in mice, showing that the Paneth cells, which are in the crypts in the intestine and are the niche for the stem cells there, and are very important in secreting antimicrobial peptides and so on, they undergo a process called secretory autophagy, which is associated with lysozyme. And they do it particularly when there's intracellular stress from bacterial infection, although it's actually mediated by immune cell effectors. And we just wondered if we might be seeing something like that here. So what we did was to look at what was happening to lysozyme. And indeed, what we find is that with short-term rapamycin treatment, we see an increase in lysozyme in these intestinal cells, and that increase depends on this LMAN5, but also our friend ATG5, which is one of the 2 ATGs that is clearly involved in the secretory autophagy pathway. So it looks as though what we've got here is a non-standard autophagy pathway that's induced normally in response to cellular stress, but is showing this long-term memory effect. Of course, what we really care about is what goes on in mammals, so we've started to look at this in mice, where we've done a very similar experiment where we fed them the drug just for the first 3 months of adulthood, so starting at 3 months when they're mature. And that we then look at a year of age, so when they're starting to be middle-aged, at some of the things that are happening in the gut or as a consequence of the gut. So on the right here, we've got lipopolysaccharide binding protein, which is a measure of leakage of bacterial products from the gut in mice. And you can see that its expression increases with age in our mice. We're going at the 12-month point. And you can see on the right the effect of chronic rapamycin treatment and previous rapamycin treatment. So we're 6 months later now. And they're equivalent. There's no difference between them. There's a reduction in this leakage. And that is associated with an improvement in the tight junctions in the gut, which may well be relevant to the leakage. So again, if we do the brief treatment early in adulthood, what you can see is the age-dependent loss of tight junctions on the left. It's rescued by rapamycin, and just as much by rapamycin that they now haven't seen for 6 months. So there's a real strong memory effect here. And we've looked at several other gut characteristics that change with age. So cells particularly show a lot of age-related changes, and we see rescue there. We see this change in lysozyme, in which the secretory autophagy, and we also see an increase in a lysosomal monosidase. So it does look as though similar things are going on here. So to conclude, we've got this brief treatment or brief induction of macroautophagy and the long-term effect. We see very similar in mice. We have lysosomal monosidase, and they and HG5 are causal in Drosophila and are associated with increased lysosomes and expression of lysozyme. We see punctate lysosomes and MAN2-positive punctate in intestinal crypts in mice. And I think generally the importance of the intestine may be being greatly underestimated. So, thank you very much for your attention.
Thank you.
Sorry, too long.
Thank you. Thank you so much, Linda. Fantastic talk. Do we have any questions? We have one down in the back. Where is Brenna?
Great talk. So I have 2 questions. So do you know...
Ask one. Thank you.
Just one?
We don't have much time. This is on a schedule. One question.
Okay. So do you have any idea like what is driving the memory changes? So is it like rapamycin is affecting somehow the histone landscape or methylation landscape, or how does this thing go on for so long?
I've just changed the slide deliberately. There's a real extraordinary complexity in belt and braces system going on here. So we've got regular macroautophagy, clearly important. We've now got the secretory autophagy. This is a paper from Yuxuan, who's here. If you feed flies rapamycin or mice rapamycin, you see a big change in chromatin. You see hugely increased expression of histones, changes in chromatin, and a non-canonical transcriptional induction of autophagy in Drosophila through a gene called blue cheese. And really this whole system seems to be geared to using multiple different cellular processes to induce autophagy-like processes, I suspect in response to cellular stress, in this case, the cellular stress that occurs during aging. And I think that that's the nexus that we're looking at, how it's clicked into an alternative stable state by rapamycin. I think we're still not clear about. This chromatin story is not part of the memory. It's an acute effect.
Great, thank you so much. We have one more question from Slack. This is from Jennifer Eliseve, who's, I guess, also in the house, I think, somewhere. But she's writing, thank you for your very interesting talk. People working in the microbiome field and notice there's a difference in colonization protocols around the time that you're doing the early RAPA treatment. Is that early RAPA impacting the immune system in the gut and tolerization to bugs introduced at that time?
I think if you wait till tomorrow, you will hear a talk from Pinksy about the S6 kinase that will illuminate you on that point, and I don't want to steal his thunder.
Okay, that's great. Well, thank you so much, Linda. Fantastic talk.
Thank you.